Thank you. I'm Taline Kazandjian. I'm from the Centre for Snakebite Research and Interventions. And as was said, I'm here to talk about some of my work on the influence of defensive venom spitting on snake venom evolution in spitting cobras, which was published in Science at the beginning of this year. So when we're looking at venom evolution, we first of all want to ask what generates venom variation upon which evolution acts. And in snakes, this evidence has shown that the factors involved tend to be diet, climate, age, and sex. And these all contribute to variation that we see across species and across individuals of the same species in snakes. However, the main factor of venom that influences venom variation is often considered to be diet, because many of these other factors can be in some way or another linked to diet. Obviously, in different climates, you can have different prey niches. Different ages of snakes will also have different prey niches, mainly because of size. And in some sexes, or in some species of snakes that have sexual dimorphism, such as this Wagler's pit viper, where you can see this small male on top of this very large female, sex differences could also be related to the different prey niches that come with size. However, one factor that isn't often considered is defense. Now, obviously, this isn't a venomous snake. This is just a python and a man regretting some more immediate life choices, but I think it illustrates the point quite nicely in that snakes can also use their venoms defensively through biting. But this isn't largely considered to be a huge factor in snake venom evolution, because snakes use their venom primarily for prey capture. Now, cobras make the ideal study species when we want to study the potential influence of defense on venom evolution, because as you can see, they have this purely defensive function of their venom in some of the species, where they eject their venom as a spray into the eyes of an aggressor. And these are rightly known as spitting cobras because they spit their venom. And this is not intrinsically linked to its effectiveness as a predatory tool. It is purely defensive in this instance. The way they are able to do this is in non-spitting cobras, as with many elapids, they have these elongate side-facing orifices to their fangs, whereas in spitting cobras, they have these very small front-facing orifices, which increases the pressure at which you can eject venom through the fang. And this spitting kind of phenomenon has evolved 3 times in the elapids in this very closely related group of animals, once in the African spitting cobras, once in the Asian spitting cobras, and once in the wrinkles, also known as Hemacartus hemacartus. And these spitting venom is thus far it's the only purely defensive use of venom that we know of in snakes. So for this study, it took a large variety of non-spitting and spitting cobras from Africa and Asia. And we also looked at a couple of very closely related outgroups, the desert black snake, Walternesia aegyptia, and the shield-nosed snake, Aespilaps scutatus. And for this project, I had 3 main questions in mind at the start. One is simply, does venom composition differ between spitting and non-spitting cobras? Does the evolution of this defensive phenomenon influence the composition of underlying venom? And one thing that suggests that it might is that in bites from spitting cobras, we know that there is clinical differences. We see from African spitting cobras particularly, they tend to have quite cytotoxic symptoms when people come in presenting with the bites from them, whereas non-spitting cobras in Africa tend to have quite neurotoxic venoms. Second question is, if this venom composition differs between spitting and non-spitting cobras, then in these 3 independent lineages, does this composition differ, or have they all converged upon a single form? And lastly, could evolution have driven venom composition designed or driven by defense rather than predation? And the first level we looked at when answering this question was the transcriptome of the venom gland. You could look at the DNA of a snake, but the venom gland is the organ specifically designed to produce toxins. And so by looking at the translated RNA, you are looking at the most functionally relevant genes to the snake. So to do this, we dissected some venom glands, sequenced them on Illumina, and basically came out with this spreadsheet of information, which gives you an idea of expression, the sequence, and what blast to go is predicted as their description. And the overview of this transcriptome is that the majority of cobras, their venoms are mainly composed of things called 3-finger toxin or 3-FTX, which are generally neurotoxic in function. However, a large number of these are what we call cytotoxic 3-finger toxins, or sometimes cardiotoxins. And these, as the name suggests, tend to have a more cytotoxic effect than a neurotoxic effect. A lot of the cobras also have these things called phospholipase A2s in quite high expression. And at face value, it looked like these spitting cobras might have more of these PLA2s and non-spitting cobras at the transcriptome level, but when I performed some basic statistics on this, it didn't really show any major differences in either of these two main venom components. So we decided to look at the next level, which is the proteomics level, because the transcriptome, while great, can't tell you everything. It doesn't take into account post-translational modifications and folding of protein, the eventual proteins. So some colleagues our colleagues Daniel Petras and Juan Calvete took some of our venoms that we extracted and ran them through a lipid chromatography mass spectrometry machine, and eventually came out with a similar sort of output with the transcriptome, where you get a percentage abundance value for each protein, an amino acid sequence, and a predicted sequence description. Now, this picture is very similar, with one key difference being that the Asian spitting cobras at this stage seem to have really massively upregulated their phospholipase A2s. But it's a similar picture in that cobras tend to have quite a high abundance of 3-finger toxins, especially these cytotoxic ones. The only major difference at this point is that spitting cobras have a significantly higher amount of phospholipase A2s than their non-spitting counterparts. And now, non-spitting cobras have a significantly higher abundance of 3-finger toxins, but this isn't due to either the cytotoxins or the other 3-finger toxins. So I think this is just because there is a significantly higher PLA2 abundance in spitting cobras. So non-spitting cobras are obviously going to have more of the 3-finger toxins. There we go. Now, base statistics is great, but you want to generally try and take phylogenetics into account, because we don't know whether what we're seeing is because of is an ancestral trait, or whether it's genuinely because it's associated with a factor that we're interested in. So I reconstructed the characteristic over the species tree of the cobras and close relatives, kindly provided by Wolfgang Worster, and then did what was called phylogenetic generalized least squares to basically take into account of, given this species tree, is the difference we see in spitting and non-spitting cobras in terms of 3-finger toxin abundance still significant? And that analysis said yes. So I again split it down into cytotoxins and other 3-finger toxins, but again we're seeing that there's no significant difference here. So I looked at the PLA2s, and again PGLS says there is a significant difference in the PLA2 abundance between spitting and non-spitting cobras, and there's also a significant difference when we looked at PLA2 enzymatic activity in the functional bioassays. So what I'm thinking is there's a significantly higher amount, as I said previously, there's a significantly higher amount of 3-finger toxins in non-spitting cobras, simply because spitting cobras have a higher amount of this secondary toxin, this PLA2 toxin. So this answers our first question quite nicely. Does venom composition differ between spitting and non-spitting cobras? And yes, spitting cobras appear to have increased the amount of PLA2s in their toxins, in their venoms. So to look at our second question, I decided to do a little more deep analysis into kind of sequence differences between the cobra lineages. So I did what was called principal coordinate analysis, which looks and sounds quite scary on first glance, so I thought I'd break it down a little bit. Essentially, you take all of the amino acid sequences and you do a pairwise comparison, which generates this table of how different each sequence is to every other sequence. And then in R, you can convert this into a matrix and perform the analysis that reduces it down into these two kind of spatial components, which you can then make a nice plot out of. So in terms of all of the toxin proteins in cobra venoms, the spitting cobra lineages actually separate out quite nicely into these three main groups. You have the Asian spitting cobras, the African spitting cobras, and Hemacartus right here, and the non-spitting cobras kind of segregate all as one cluster, with an exception of Philippinensis, and I think that's because Philippinensis has very notoriously neurotoxic venom. So it could be that in terms of amino acid sequence, there is something in the structure of one of its toxins that is called, or more of its toxins, that is causing it to its venom to behave more similarly to venoms of non-spitting cobras. So in order to investigate these differences between these spitting cobra lineages more thoroughly, I decided to look again at the main components of the venom, one being the cytotoxins and one being the PLA2s. And here I sized each data point by their relative abundance in the proteome to get an idea of where the most abundant proteins are in terms of similarity to each other. And if you look at the cytotoxins, all of the major cytotoxins in the venom are really highly conserved. Whereas if you look at the PLA2s, it actually splits out into these sort of three groups again. But interestingly enough, the Asian cobras all have very similar structures in terms of their PLA2s. The African and the Hemacartus have very similar structures in terms of their PLA2s, and then the African non-spitting cobras sort of linger in the middle. And to look more deeply at this, I basically did a comparison to see which regions of these sequences were conserved. And the main differences I could see between the spitting lineages is that there's in the African spitters and Hemacartus, there seems to be this kind of deletion here. And there's also there is a substitution in this position between the African spitters, the Hemacartus, and the Asian cobras. Then I decided to see what this could mean in terms of how it affects the protein. So I looked at hydrophobicity, charge, and conservation on the actual 3D structure. And it looks like these regions of variability are potentially happening in areas that are fairly hydrophobic. But unfortunately, I am not a biochemist, so I don't know exactly what that could mean, just that this is occurring. So this does partially answer our second question, which is, does venom composition differ between independently evolved spitting cobra groups? And yes, though spitting cobras have converged upon increasing the abundance of PLA2s in their proteome, the structure of these PLA2s is actually very different. Which leads us on to our third question, which is, could defense be driving the evolution of these venom proteomes in cobras? And to look at that, we want to look at the toxin in question that is of significance, which in this case are these PLA2s. Now, unfortunately, PLA2s are hugely multifunctional, which is annoying to try and figure out what could be causing it to become upregulated, what function is useful. And when we're looking at defense, the most useful things that we would think of would be cytotoxicity, because if the cells in your eyes are being destroyed by this cobra venom, that's unpleasant and will cause you to not be able to see very well. And also pain, which tends to cause an immediate response away from whatever has inflicted pain at you. So Yuri and Michael did some irritation scores to look at the cytotoxicity of cobra venoms, and what they did is they basically put cobra venoms onto hen's egg membranes and measured the amount of vascular irritation. And it seems that most cobras cause some degree of vascular irritation. And when I looked at when I performed analysis on this data, there really wasn't any difference between spitting and non-spitting cobras. But then Ivan, Irina, and Sam did what was called a calcium influx assay to look at our other hypothesis, which is pain. Now, pain is very, very, very hard to measure. You can't just jab a mouse and ask if it's okay. So what they did is they got these isolated trimaginal mouse neurons, which are the nerves that innervate your face, and they applied cobra venom to them and measured the the EC50 values of these venoms, which is the amount that is required to cause at least 50% of the activity, which in this case is calcium influx into these neurons, which stimulates these neurons. And when you plot this over the species tree, you can see that there's a very, very clear pattern that spitting cobras cause massively increased calcium influx into these neurons compared with non-spitting cobras. And this is highly significant. So to further investigate what toxins could be causing this, they isolated the cytotoxins, known here as cardiotoxins, and the PLA2s. Now, when I thought that PLA2s on their own, because they are increased in abundance in spitting cobras, would obviously cause this pain activity. However, when you add them on their own, there isn't actually a response. It's this increased calcium influx response is actually caused by cardiotoxins or cytotoxins, which are present in all cobra venoms. However, if you add PLA2s to these cytotoxins, it causes this response to increase massively. And what this tells us is that cytotoxins are responsible for inflicting this pain, whereas PLA2s are pain potentiators. And this is quite exciting because it means that you have this inherent molecular defense that is available in all cobras, which is these cytotoxins, plus the behavioral defense that is a hooding, raising of the body off the ground, neck raising, striking. And this combined facilitated the evolution of defensive venom spitting. And then once venom spitting evolves, upregulation of PLA2s is supported because these PLA2s are increasing the effectiveness of each venom droplet that hits your eye, and every little helps. So this sort of answers our third question. Could evolution have driven venom composition in spitting cobras designed for defense? And the evidence suggests that yes, it has, because spitting cobra venoms stimulate more activity in these neurons than non-spitting cobra venoms because they have these PLA2s that highly increase the activity of the cytotoxins. However, after all this, there was still kind of one overarching question, and that is, we know that PLA2s are increased in spitting cobra venoms, and this is probably because it causes more pain when venom hits the eye. But why would this evolve, and why would spitting evolve in these closely related group of Afro-Asian lapids? What would cause something like this to evolve? And when you're looking at this, you want to ask, well, what is a strong enough driving force to have driven such an unusual combination of behaviors? And one is potentially, is it an anti-trampling defense? Is it a way to tell large herd animals, please don't step on me? And I think this is unlikely because you still see spitting cobras in forested areas where a lot of these herding ungulates are found in savanna brushlands. You do get solitary ungulates in rainforests, but I feel like they would be much less likely to trample on snakes than if they were a large congregation of animals. And also, ungulates don't tend to be that visually oriented. You can see these animals, they have large ears and they have large noses, and that's because they use them. They don't tend to, if you took out both of their eyes, it's probably not that much of a hassle to them compared to if you took out their nose and ears. So it's possible, but I would think it unlikely. A second culprit is birds. Birds are a common predator of snakes. Is it possible that cobras have evolved a spitting defense against birds, which are visually acute and visually oriented predators? And again, I think this is unlikely. For one thing, many birds attack from the sky, so I don't think that would give any snake time to react, yet alone turn around, hood up and spit at a bird before it is taken down. And for those that don't fly, well, clearly enough snakes are being killed by birds that if it has evolved as an anti-bird defense, it's not working. A lot of cobras get eaten by birds. So this leads us to our third and what I feel is the most compelling culprit for promoting the evolution of defensive venom spitting, and that is bipedal hominids. Now, obviously, there aren't any kind of early ancestors of humans alive today, but even if you just look at apes now, chimps, they can recognize snakes on sight. Some of them will even kill snakes as a preemptive measure or out of fear. They are visually reliant, and they are a long-distance threat. Even chimps can throw things like sticks and rocks, and obviously early ancestors of humans would have had things that are much more threatening, like spears. So there isn't a huge amount of evidence to substantiate this, but if you look at the evolution of spitting and you overlay some significant evolutionary events in the emergence of bipedal hominids and of a man, you can see that the divergence of Homo from Pan, which is chimpanzees and bonobos, kind of coincides with the emergence of spitting in these two African groups, which is Hemacartus and the African spitting cobras. And then the arrival of the Homo genus in Asia correlates with the emergence of spitting in Asian cobras. So, though it's not direct evidence, I think it's very compelling. Now, after all this, where is the next direction to take this? And one thing because we now know that defense can be an important factor in venom evolution, as well, particularly, specifically in cobras, but potentially in other snakes we haven't looked at, which begs the question of if defense is a driver of venom evolution in cobras and potentially other snakes, does it conflict with other factors of evolution, especially things like what we consider to be the most dominant influence of venom evolution, which is diet. Now, I have some very, very, very preliminary analysis, which I've been doing on Naia nigricollis. Essentially, I have Naia nigricollis from Tanzania, Togo, and Nigeria, and I had a little bit of a look in some of the literature, which was basically I looked at the literature that was comparing the proteomes of non-spitting cobra species from different regions. And I decided to just throw that all into a principal component analysis and then see where it lands. And interestingly enough, these Naia nigricollis seem to have fairly more conserved venom composition at the proteomics level, even though in terms of distance on the global map, they're actually quite far away from each other, some of these populations, compared to things like especially Naia, where they have a hugely diverse venom variation. And even some more easternly Asian populations of Nyakuthia, which are fairly closely located to each other compared to Tanzania and Nigeria, they have a very diverse venom composition. And obviously, this doesn't take into account things like topography, but in terms of just simple different distance, where you might expect different prey niches would highly influence or cause different venom variation in these species, we're just not really seeing that much in Naia nigricollis. And is it because this defensive evolution is causing the venoms to focus on upregulating PLA2s rather than focusing on diversity for prey capture, which obviously isn't a question that I can answer right now, but I think would be an interesting one to leave off on. So I have a huge amount of people to thank. As Anna said, I think it was 27 authors on the paper that I was working on. So I'll leave this up here for a few moments. But yes, thank you to all my fellow co-authors and everyone who's helped me on this massive journey. And also thank you for listening.